Good evening, everyone, and welcome to the Smithsonian National Air and Space Museum's Exploring Space Lecture Series for 2021. I'm Ross Irwin, geologist and chair of the museum's Center for Earth and Planetary Studies, where we do original research on the planets and moons of the solar system. This year's Exploring Space Lectures are focused on missions that visit and sample other worlds, the planets Mars and Venus, Saturn's largest moon Titan, and the asteroid Bennu. Landing on these worlds has given us a clearer idea of what they're made of and how they evolved. It also has turned these points of light in the sky into worlds in many ways like our own. It's my great pleasure to introduce our speaker tonight, Dr. Moo Cooper, the lead Planetary Protection Engineer for the Mars 2020 Perseverance Rover at NASA's Jet Propulsion Laboratory. She plays a critical role in ensuring that any evidence that we may find of life on Mars is from there and not something that we brought with us. Perseverance will study the surface of Mars, collect samples for later return to Earth, and help us plan for future human exploration. Dr. Cooper received a BA in physics from Hampton University in 2006, followed by a master's and PhD in mechanical engineering with a concentration in thermal fluid sciences from Drexel University. Her dissertation involved non-equilibrium plasma sterilization of spacecraft materials. She now works for JPL's Planetary Protection Group. She's received awards including the NASA Early Career Public Achievement Medal, the Charles Alachi Award for Exceptional Early Career Achievement, and JPL Voyager Awards for Technical Leadership. After Dr. Cooper speaks, we'll have time for questions and answers, so please feel free to submit your questions in the chat. We'll answer as many as we can. Afterward, you can visit the National Air and Space Museum's website for more information on the upcoming Exploring Space Lectures. I'm grateful to our sponsors, Aerojet, Rocketdyne, and United Launch Alliance, for their continued support of this series. And thanks again to all of you for joining us tonight for this fascinating look at Mars 2020 and the importance of planetary protection.
Thank you so much, Ross, for that wonderful intro. And of course, we start with technical difficulties because this is how it goes. Thank you so much, Ross, for that great introduction. And I hope by the end of this lecture that you all are planetary protection experts and that you can also be honorary spacecraft cleanliness gurus. So let's jump into the talk. So this is a schematic of the Mars 2020 mission, which I'm sure you're all very familiar with. As you see on the left, there's a schematic of the actual rover itself. And this rover is amazing. It is essentially a lab on wheels. And the great thing about a lab on wheels is it has fantastic capabilities, such as, hold on, there's an echo down here. It has awesome capabilities. For example, it is able to collect samples on the surface of Mars, which is amazing. It is able to look at the geology on the surface of Mars and just do incredible things. The only disadvantage of having a lab on wheels that is 34 million miles away is that if anything goes wrong, like my technical difficulties today, you can't just go and fix it. It's so far away, you can't do repairs. So for that reason, one of in my opinion, the most exciting things about this mission is that we're going to be collecting samples and sending them back to Earth so that we can really interrogate them with the best instruments that we have here on Earth. You can imagine too that if you are taking instruments and you're miniaturizing it, there's a lot of limitations that you get from shrinking different instruments. So that's why bringing these samples back is so important. So I'll talk a little bit more about what it's going to do and some of its other scientific goals, and let you know how planetary protection fits into this entire grand scheme. The other extremely unique and cool thing about this mission is it has a sidekick, Ingenuity. And Ingenuity is going to be actually taking its first flight relatively soon. And it's a tech demonstration that will allow us to understand, number one, can we fly helicopters on another planet? And anything else beyond that is just gravy. So the goal is hopefully in the next few days that we can see some incredible footage here. It's a huge engineering feat to get a helicopter to fly on Mars, you have about 1% of the atmosphere. And so you have to imagine how difficult it is to generate that lift that you need to fly any type of aircraft. So we're all going to be holding onto our seats, and we're just going to be excited about this next milestone. Here's an overview of the entire mission. This, as you can see on the left side here of your screen, it launched in July of 2020 on time, and in fact, a few milliseconds early. We spent about 7 months going through a cruise phase to get to Mars, and we landed very recently on the surface, and we are now starting the operations phase, and that should last about 1.5 Martian years. The landing site that we are currently at is called Jezreel Crater, and you can see where the location of Jezreel Crater is with respect to all of the other landing sites for our prior rovers. The location, Jezreel Crater, is so important because, among many other things, it used to be an ancient lake. And at the bottom of the ancient lake, so imagine, for example, you go outside, you find your nearest lake or river, and if you just scoop the sediment at the bottom of this lake or river, and you take that sample and you put it under a microscope, you will see a high quantity and diversity of microorganisms. So you can imagine that if you picked a site where it used to have water, it used to be a lake, you can really have a really high chance of actually finding ancient signs of Martian life. So it looked like what you see on your screen about 3.5 to 4 billion years ago. And this is what it looks like today. You can see the beautiful delta here on your screen. And this is where you're going to find just a lot of interesting possible potential fossilized Martian microbes. So we're all crossing our fingers and hoping that we get to finally discover that. Here is a quick tour of the rover. I highly encourage you to go on NASA's website. I have the link below to be able to actually explore in 3D the actual rover yourself. You can see there are several components like the coring arm, the coring drill, which is up here on the arm. And underneath here, you can see a view of the belly pan. This over here is where the ejectable belly pan was. And you saw that it recently ejected itself. And then back here is where the helicopter is mounted underneath the rover. So you can go online and do that really fun 3D tour. There are a few things that I wanted to highlight on the rover, and this is one in particular. It's a calibration target for the Sherlock instrument. And the piece that I want to point out, or the pieces that I want to point, is on the left side of your screen here. These are pieces of an astronaut's suit. It's what is used today typically to build astronaut spacesuits. And the hope is to take a look at this and see how it degrades over time so that we're able to prepare for humans to go to Mars in the future. For those sleuths who have already figured this out, I'm sure this is not going to be a surprise for you. But if you look at this particular calibration target here, this is a piece of visor material that usually astronauts use. And if you zoom in, there's the address of Sherlock Holmes, the fictional character, etched on this visor material. So if you did not know that already, take a look, go on NASA's website, and you can see a much better view of the etching. So back to the sampling and caching part, just a quick overview. We're going to collect about we have 43 tubes. About 20 of them are going to come back, hopefully. And so hopefully we'll target around 20 samples that we'll collect, return back to Earth, not to Mars. And that will allow us to interrogate those samples with the best instruments that the Earth has to offer. So these sample tubes are really fantastic. I'll show you a few pictures of the tubes loaded into the fixture before it gets loaded into the belly of the rover. But it was a huge feat of engineering skills that thousands of people had to come together and make this work. So after we collect those samples, it's periodically going to be dropped off along the surface of Mars as we're doing our exploration. So you can see that video here is just a notional representation of how it's going to be dropped off. And then eventually a future mission will come and pick those samples up and bring them back to Earth. And that whole retrieval part is actually going to be done in collaboration with the European Space Agency. So this sample return venture is a huge feat, and it's so great that we get to not only symbolically, but truly unite with people around the world to make this happen. So this is just a cool cartoon that shows how we sent the Mars 2020 rover, Perseverance, onto the surface of Mars. It is going to collect its samples. In parallel, NASA and the European Space Agency will be working on orbiters and landers that will collect and fetch these samples and then bring them back to Earth. So the goal is hopefully to get this done within the next 10 or so years. So you've heard this overview. I'm sure many of you have paid attention to all of the fantastic things that are happening right now on Mars. But how does planetary protection fit in? So planetary protection is a discipline that has two different goals: to protect the planet that we're trying to explore, so Mars in this situation, from any inadvertent Earth microbial contamination. So we don't want to go there and discover life and come to find out it's actually a microbial hitchhiker from the Earth. And the second thread of planetary protection is once we bring samples back, we need to make sure before we bring them back that it is able to be brought back in a safe way so that we can protect humans in our own biosphere from any inadvertent contamination. So planetary protection is twofold. And today I'll be talking to you more about the first part, the forward planetary protection, and to show you how we keep Mars clean. So this is a really busy chart, but the chart kind of gives you an idea of how we clean and keep the spacecraft clean. So on the left side of your screen, so the cleaning part is quite easy, but keeping it clean is very difficult. So on the left side, you can see ways that we keep the hardware clean. So you can see, for example, we wipe the surfaces, the critical surfaces that could be possibly recontaminated with isopropyl alcohol. And this, just like what you do at home on your own surfaces, especially with this global pandemic happening, I'm sure you're constantly wiping down surfaces and cleaning your hands and also wiping down surfaces with alcohol. And we do the exact same thing for spacecraft. Other ways we keep it clean is the vast majority of the components on the spacecraft actually have gone through a high temperature process. For example, so here's a picture of what we call the DVT. It's the dispenser. It's the volume assessment probe and a set of tube assemblies. So the seal dispenser, volume assessment probe, and the tube assemblies are in this little triangular feature. And it was loaded all together into this chamber that you see here. And it was exposed to 150 degrees Celsius for 29 hours. And that was able to clean these surfaces to the cleanest level that we have ever sent any spacecraft into out to Mars. So it's pretty incredible the lengths that the team has gone to ensure that not only is the spacecraft clean, but the parts that are going to touch the Martian soil are extremely clean. And the last thing that we do, or one of the last of the three examples that I have, because we do many more things, is we also autoclave a lot of the tools that touch the critical components on the spacecraft. So autoclave, that word may sound familiar to you because when you go to the doctor's office or the dentist's office and they take out the sterile tools, there's usually a pouch that they open. And that's because that's an autoclave pouch. They put their tools in there, they run it through the autoclave, and then whatever's inside stays sterile. And we do the exact same thing for all of the critical hardware that will touch the surface of Mars. We make sure we touch it with only sterile tools. And then on the right side of your screen, there's the motto, trust but verify. And that's what we do as well for the spacecraft. We, at least in planetary protection, are very cautious to make sure that, yes, we have our assumptions of how clean it was when it left the cleaning process, but we have to make sure that we have kept that level of cleanliness. And the best way to assure that is to sample. So on the screen, you can see this is the belly of the rover, and this is the where the ejectable belly pan was as of a few days ago before it was deployed. This picture was taken right before the ejectable belly pan was placed on the bottom of the rover for the very last time until right a few days ago when it was finally deployed on the surface. So taking these samples right before closeout allows us to ensure, yes, everything was integrated in a clean way, and we can ensure that we can close this up and continue to move on. An interesting sight in this photo as well, you can see this black structure here. That is the helicopter debris shield, and that is what just deployed yesterday onto the surface of Mars. So you can get a sense of scale of how large or small these particular surfaces are compared to a human. So the biggest thing, the biggest threat in our clean room that we have against spacecraft cleanliness are humans. Humans have more microbes in and on our body than we have human cells. So that's why in all of these pictures, you see that I'm wearing a bunny suit, and everybody who has to interact with the spacecraft, they have to wear these protective garments to keep them separated from the spacecraft. And the second biggest thing, the second biggest threat that we have against spacecraft cleanliness is also the environment. So wearing these suits also keeps the environment away from our spacecraft as we build it. So planetary protection is woven into every single detail of the entire mission, all the way from when we're designing the actual tubes and tube assemblies, all the way up to building it and implementing the day-to-day operations. Just to give you an example of one of the things that we do that has an influence with planetary protection, I have this picture here in front of you. It is the rover turned upside down, and it's in the stow position, meaning the legs are raised. This is the position that it was in during that 7-month cruise. And through a lot of entry, descent, and landing before the wheels dropped down and it softly landed on the surface. So this stow position that it's in, it was essentially on this spin table, which you can't see in the frame, just to make sure that the calculations that we have for the center of gravity is correct. So while it's on the spin table, and since they took this picture, I wanted to point out a few features that are there just because planetary protection exists. So here you can see what is highlighted in yellow are planetary protection specific things that we've done. So these wheels that you see, not all of them are highlighted, but the wheels that you see are covered in a film called Amerstat. And this film, this barrier, is able to protect these wheels since it will directly touch the surface of Mars from any recontamination. So these covers stayed on till the very last moment that we could access the wheels. And then they were taken off and we archived those actual materials. But that allowed the wheels to stay microbially clean. A few other features you can see here in the middle, this is the bit carousel. It's the part that you see that's kind of sticking up on the top deck of the rover. And this bit carousel is, let me just plug in there's a low battery symbol, which I'm gonna duck under my desk one second. There we go. So that bit carousel that you see in the middle is an actual biobarrier. And this biobarrier means it's sealed off from the external environment And the only thing that separates the external environment and the internal environment is a HEPA filter as far as the transmission of any gas or molecules. So this HEPA filter it allows us to keep the internal components, the bits, the abrasion tool, the coring bit, and the regolith bit, extremely clean, even through this assembly process. So that's another planetary protection feature that is on the rover. And then, of course, our most important coring drill here on the left side, these stabilizers that touch the Martian soil are covered until the very last minute so that we can keep those interfacing surfaces extremely clean. So for planetary protection, if you're wondering, well, what do you do on a day-to-day basis? Well, when we take these samples, so first we make sure the spacecraft is clean, then we take our samples, and after we take our samples, we actually bring those swabs or wipes back to the lab, and we go through a process called the NASA standard assay. And this assay looks for a particular kind of microorganism that could actually withstand the vacuum of space and the very harsh environments that you go through between Earth and Mars. And we target those microbes because those are the hardiest that we could find here on Earth. And so we want to make sure that we limit the quantities of those particular microorganisms. And so once we look in our lab for these microbes, we grow them on Petri dishes. And based on how many colonies grow on our Petri dishes, then we can back calculate and understand how clean the spacecraft is. So these specific microbes that we're looking at, they're called bacterial endospores. And these endospores can stay dormant for millions of years, sometimes tens of millions of years, until the right conditions come about, and then they germinate and grow. So that's why we target these particular kinds of microbes, because they can essentially find a nice, warm, balmy environment and then start colonizing or growing in that particular environment. So that's what we do not want to happen. So in addition to sampling, growing, and identifying these spore form, these bacterial endospores, we also, after we identify them, archive them. We have an archive that it goes all the way back to Viking, when planetary protection was first used on spacecraft. And we have a freezer with all of the microbes that we've ever found, so that one day if we wanted to do tests on how hardy they are or why they may have survived all of the cleaning, that is where we access these extremophiles. We also take samples and we look at the DNA. So you can imagine when you do this process up here, only a certain amount of microbes, 0.1% of microorganisms, can grow on a Petri dish. And then in the particular Petri dish that you have, if they don't like that particular food, they may not grow effectively. So what we also do is we take DNA, because DNA doesn't matter if it can grow on the Petri dish or not, the DNA will still be there. And so we extract the DNA and we actually archive, we save the DNA for a future sequencer, a future technology that might be able to detect small trace amounts better. And then we also sequence it ourselves. So we essentially 23andMe our samples and we can identify what specifically is there. And based on this list, in the case of Mars 2020, we have a catalog of all of the possible forms of contamination, so that in about a decade or so, when we get these samples back, we have something to compare it with. And hopefully we don't find any contamination, but just in case, just to dot our I's and cross our T's, we have this list to compare against anything that we find. So we are sampling and cleaning and verifying at all times throughout the entire mission. Our first sample, I remember, was in 2014. It was the descent stage. And so since 2014 till before we launched in July of 2020, we collected 13,042 swabs. We collected 3,521 wipes. 318 air samples, because you can imagine as you talk and breathe in the air, you're spreading different particles. And of course, in the clean room, we are fully garmented, so that shouldn't happen as much. But microbes are in the air as well, so we collect air samples. And we also took 1,122 DNA samples. So we were very busy throughout the entire duration of the mission. And the cleanliness levels that we achieved when you add up all of the results at the end of the day was significantly lower than what our requirement was. There's an international treaty called the Outer Space Treaty that the United States is a signatory to. And so that dictates how the maximum level of microbes that we have on our entire spacecraft. So that number for all of the parts that land on the surface of Mars is 300,000 spores. And against that requirement, everything that landed on the surface of Mars, if you add that all up based on our samples, was actually far cleaner. So 3.86 times 10 to the 4. So about an order of magnitude cleaner than what our requirements are. So 87% cleaner. So, which is a huge achievement really for the engineers and the folks that assembled the spacecraft on schedule and with the right cleanliness protocols. And for the entire spacecraft, so you can imagine, we're not only focused on the rover and the descent stage and the cruise stage. We also want to know, you can see on your upper right-hand corner of the screen, we have a launch vehicle. In fact, the launch vehicle through ULA. And we have to take samples of this fairing to make sure that it is also clean enough. And that also came in far cleaner than what we were expecting. So the total, if you add everything up on your entire spacecraft, the fairing, the aeroshell, all the parts that could possibly recontaminate the bits that are going to land on the surface of Mars, the total maximum amount of contamination we can have is 500,000 spores. And we were at about 37,000 spores. So we had about a 25% margin on that. Oh, sorry, 373,000 spores. So we had about a 25% margin on our requirement, which is pretty incredible. So with all of that, sampling all the way until the end, we got to this final milestone, the launch. It was an incredible day. I'm sure you were all watching from your homes and your workplaces. And we finally landed. The 7-month cruise has ended. We landed, and this is just a recreation of the rover's first drive, of Perseverance's first drive. The recreation is found actually on the NASA website. But it's amazing. The first thing that you do when you land is you make sure that everything checks out all right. So they did all the checkouts. They moved the rover around. They were able to see that indeed everything worked. The mobility is fine. We're looking good so far. And so you saw it kind of traveled up. It did a spin. It backed up, and that kind of tracks. You can see this picture. One of the earlier pictures of it driving up, it's turning around and backing up. So it's pretty cool to see these images from Mars and how it tracks with what the rover is actually doing for the day. And I'm sure you've seen the latest news with the belly pan drop, or sorry, not the belly pan, the debris shield drop, and that revealed the helicopter that you see underneath. And soon, it sounds like we're going to be days away from finding the location where they're actually going to deploy the helicopter, which is exciting. And we hope that the helicopter will give a fantastic technology demonstration to show that, yes, indeed, we can fly a rotorcraft on another planet. The cool thing that was finally revealed today, the helicopter actually has a piece of the Wright brothers' airplane from its first flight. And it's amazing that it's going to make history yet again. And the way planetary protection fits in is we actually had to sterilize that piece of material because for the reasons that we talked about all throughout this lecture, we can't bring any microbial hitchhikers that may contaminate the critical parts of our spacecraft. So that piece of Wright Brothers material is actually a sterile piece of airplane that it brought with it. So ultimately, after we get through the fun helicopter flights and the amazing engineering and science that we can get from that, I'm truly excited about us collecting samples and eventually bringing them back to Earth. My test by exam will be kind of brought to fruition once we bring these samples back to Earth, and we can see if we did it in a way that was clean enough. Were we stringent enough so that whatever we find in there, we won't have extra bits of contamination? And that's what I'm really looking forward to, is looking at those samples in a decade or so. So ultimately, the goal here is to let you know about planetary protection, to have that in the back of your mind or in the front of your mind when you're looking at other missions in our solar system, missions to Europa, missions to other moons and planets where there may be some form of life, or at least the conditions could be right. And the main thing I want you to remember is to be good custodians of our universe and explore responsibly. So thank you so much for your time.
Thank you very much for that presentation. I mean, you really have taken this incredibly complex mission with all of these moving parts and condensed it down to this very understandable intersection of engineering and biology, and you've made it all work. Thank you so much. I'm Ross Irwin with the National Air and Space Museum, and I'd like to invite our viewers to submit questions in the chat. We've already received a number of them, so I'll start with those. The first one: what is NASA doing to protect Mars from litter, like the heat shield and other space junk? Now, keep in mind, you're in the Smithsonian now, and to us, that stuff is artifacts, and we would take it in a minute if we could get our hands on it.
Yeah, that's a great point. Yeah, and in fact, the artifact, the Wright brothers plane, I believe, was lent out by the Smithsonian, if I'm not correct. So yeah, of course, it's a high priority to not litter on other planets. The thing is, when the parts of the spacecraft that we have on Mars have been cleaned to a very high degree, so the aeroshell, which is composed of the heat shield and the back shell, the heat shield is extremely clean. It was baked out just through the process of assembling it, and it went through that high temperature ablation process as it was going into the atmosphere of Mars. So it's extremely clean. The back shell is clean. The descent stage is actually ridiculously clean. So the very few pieces that we put on the surface of Mars at least are fairly orderly and microbially clean.
When the sample tubes are dropped off, how can the next mission find them and pick them up? Are there concerns about the dust interfering with that process?
Yeah, it's interesting. A group of far smarter people than I came together and actually figured out that they needed to barcode the outside of the tubes. So if you see, I think I have a little 3D printed model of the tube that I painted myself, so don't judge. But the real tubes actually have lines on the outside of the tube in a way that makes it like a barcode. So they're able to identify each tube, and there's also a number, the tube number on the side. So you're able to identify which tube you're looking at and map that to the sample that you took. Also, these folks gathered together and realized that even with the winds and the dust moving around, it shouldn't be enough where it's going to be completely buried. And that's why the partnership with the European Space Agency is so important, because they're working on that fetch rover to make sure that even if there's a little bit of dust covering it, we'll be able to find it, secure it, and load it back on the rocket that'll launch it into orbit.
Are there national or international standards for ensuring that these samples that are collected on Mars are sterile, or are you all basically just setting the standard yourselves at a very high level and working to that?
Yeah, it's a mix of both. I mean, there are international requirements that the European Space Agency and NASA abide by when it comes to cleanliness and planetary protection. But how you achieve that is really forging new and interesting trails. For example, the biological, looking at the DNA when we take our samples, that was not forged in the rules, the international standards. But we felt that based on what we're doing, the science that we're trying to achieve, and what we intend to do once we bring it back to Earth, that it's important to also add sampling for DNA. So it's a little mix of both. There are international rules, but we really have to blaze a new trail to do the right thing.
And you have this opportunity when you bring these samples back to do a lessons learned and to be able to figure out what worked in the past and what hadn't.
Exactly, yeah. And even after every mission, we write actual lessons learned documents just to make sure that we can improve for the future. But also the ultimate lessons learned will be found once we bring the samples back. We have something called blanks that we're sending. It's kind of like a negative control for the scientists out there. And these negative controls are going to go through all of the environments, but a sample will not be acquired. And if we can collect a sample and compare it to this negative control, then we can see, all right, what maybe came from Earth, maybe during the process, during EDL, maybe something dislodged. We'll be able to dissociate something that came from Earth with something that actually is native to Mars.
And I love this next question. Please tell us that you fixed the wheels. I mean, I'm sorry, I should phrase that in the form of a question. Did you fix the wheels relative to the ones that were in Curiosity?
Yes, so the mobility team worked tirelessly to make sure that the wheels are now thicker. If you imagine, or if you have a phone next to you and can Google the wheels, you can see the style of the grousers. The grousers are like the treads on the wheel. They're closer together, which adds a little bit more strength and integrity to the wheel. And the wheel itself is a little thicker. So yes, the wheels should not be an issue anymore this go around.
Was your team working in a Class 10 clean room when prepping the rover, or how can you elaborate on those safety measures that you were using?
Yeah. So that's a really great question. That tells me you understand clean room terminology. So kudos to you. So no, we did not build the entire spacecraft in a Class 10 clean room. It was actually built in a Class 100,000 clean room. And that facility was still clean enough. And that's why we did a lot of sampling and monitoring, and also put folks in bunny suits, because the requirement for the spacecraft is not necessarily to be sterile, just to be extremely clean under that 300,000 and 500,000 spore level. So we were able to achieve it in this 100,000 Class 100,000 clean room. That does not go though that does not apply for all of the tubes, the seals, everything that's going to touch the spacecraft or to touch the Martian soil. This was actually built on ISO 5 flow benches, so it's far cleaner environments. And so yeah, it's a little bit of a mix, but the rover itself was in a 100K clean room.
Now, in the future, we all hope to be able to send people to Mars. And it seems to me like that is really a very different problem, because you're not going to be able to sterilize the spacecraft and kill everything on it before it launches. Yeah,
and even more importantly, we can't sterilize people, and that's okay. We aren't meant to be sterilized. So the whole framework of planetary protection is going to change once we send humans to the surface of Mars. And it's important that's why it's really important to get all of the science done now before humans have a presence on the surface of Mars, because we do introduce our own microbial ecosystem into the environment as soon as humans step foot on the surface. So yeah, that's what makes Perseverance so much more critical before we send humans to Mars. Because, of course we want to send humans to Mars.
Right.
We want to see Mars with human eyes.
Yeah, and Mars is really a tough place to be. I mean, you talk about bacteria surviving in Mars, but there's the radiation environment, there's the low air pressure, there's this huge range in temperature. Is any research being done on radiation protection for future humans on the surface of Mars?
Yes, yeah. So the calibration target that Sherlock carried with it, I focused a little bit on the visor and the cool address on that visor material. But if you look at the calibration target below where you see that circular visor cal target, you'll see a series of squares, and there are various materials that comprise an astronaut spacesuit. And so that's one of the things that we're trying to do to ensure that we understand how the radiation may degrade those materials in order to protect humans a little bit better. And also we have other elements, for example, the META instrument, which is looking at the radiation, the flux that we're getting onto our bodies. If we were to send humans to Mars, there are sensors that will allow us to measure how much radiation that a future astronaut will be exposed to. In addition to that, we're looking at weather. We have MOXIE, which is also generating oxygen from CO2, hopefully for breathable air, and also as fuel for a return journey for a future astronaut. So there are a lot of things being done, not only with the spacesuit protection side, but also just to ensure that humans are able to really survive, have oxygen, and have a way to come back.
Yeah. When you think about this sample return mission, which will follow up and collect samples, you're going to have a lander to touch down on the surface. And there's also going to have to be a rover, which will go out and collect these samples. But is NASA also preparing an additional rover mission to collect samples from other places that we might be able to pick from?
Sorry, are they preparing a rover to collect samples from other places on Mars or other places in our solar system?
Other places on Mars.
Yeah.
Are we going to pick up the samples collected by this rover, or are we going to have a choice?
Yeah, so the future mission that we're actually working with the European Space Agency is to send a fetch rover, and this fetch rover is going to collect those hopefully around 20 tubes that was acquired throughout the duration of the exploration mission. Whether or not it's going to do anything besides collecting the samples, I think is an upcoming conversation, because of course, why have one function when you can add a bunch of instruments possibly? So there are still conversations ongoing to see if it's going to possibly do other things besides collect the samples on the surface of Mars. I'm excited along with you to find out.
Yeah, absolutely. And how do these rovers navigate on the surface of Mars? There are no roads. You can maybe see some landmarks in the distance, but how do you plan these trips out from one day to the next?
Yeah, there is a fantastic group of mission planners out there, which is not my job, but their job is to essentially plan out not only on the day-to-day basis, but there have been long-term, short-term plans that have been hashed out so that we can understand what is ahead for the day and what science will be done, and just what is on the docket. So yeah, there are mission planners that have that as their day-to-day job. It's pretty cool.
We've seen an increase in the size of these rovers. I mean, the first one to operate on the surface of Mars was quite small. Mars exploration rovers had about the same size and top speed as the Galapagos tortoise. This one, we're now up to sort of VW Beetle size, but not nearly the speed of a VW Beetle. So we're not really hitting the speed bumps as we're going along here. You can kind of take this a day at a time and think a little bit about what you're doing next.
Exactly. As a very smart engineer on our team, our chief scientist says, science does not happen quickly, right? So you can design a rover that will go at top speeds, but that is not what we're trying to do. We're trying to look around us and really take everything in and collect some amazing samples to bring back to Earth. All of our Martian samples have been based on kind of luck, really. It's whatever geological process happened in the past happened, and then something impacted that piece of rock and it was ejected from Mars and somehow landed in Antarctica or in the desert, and we were able to collect those samples. So now we actually get to choose which samples come back to be analyzed. So I think it's going to be a lot of fun, and it takes time.
Yeah. And the nice thing about these samples is we know where we got them. I mean, the meteorites we pick up in Antarctica, they could have come from anywhere on the planet, and we just have no idea.
Exactly. Yeah, it's amazing to have so much metadata associated with a sample before it even gets here. with Sherlock and Watson and all our instruments, all of our cameras, we'll be able to really have a lot of information on what the rock is made out of, what morphologically the sample looks like, and what features we should be paying attention to in order to evaluate whether or not we even want to bring it back. But we're going to have so much information before it even gets here. We're very, very fortunate to be able to really choose wisely.
As much as anybody, you know how to find life in small quantities. How do you do it on Mars?
Yeah, that's a really great question. And that's actually something that we've been struggling with quite a lot in the field of planetary protection. There's a lot that we borrow from our medical brothers and sisters as far as detecting DNA and looking through various using microscopic techniques. But where we struggle is, and the difference between planetary protection and other disciplines is we have a tiny amount of samples that we're looking for. If you go, like I said, dig in your nearest lake, you'll find a huge quantity of microbes. Same if you're looking at stomach microbes, you have a huge quantity and diversity. And yeah, it makes it really difficult when you're using a lot of these methods for only 10 or 15 or 20 microbes total on your sample. So yeah, it becomes difficult when you're looking for such small amounts, but that's where the challenge and the new technology developments lie. And I think there's so much more evolution this discipline can do and is doing already, just based on those challenges of low biomass.
So in addition to being an engineer by training and trade, and also in a way a biologist, you do a lot of public outreach. And so one of the questions is, who were your role models when you were younger? And what advice do you have to people who want to follow in your footsteps?
Yeah, I've had several fantastic examples along the way and continue to accumulate role models to better myself and try to communicate better to others. But my first major role model was Carl Sagan, and watching his Cosmos series just changed my life and changed my world and allowed me to realize that you can have the most complex concepts and still be able to translate it so a young child who doesn't quite know what they're going to do with their life has figured it out and knew and turned out to be, not quite an astrophysicist, but someone that was inspired by an astrophysicist. So yeah, I would say my advice to you is just continue to learn from people who you feel are great communicators, if you would like to do this as well. And yeah, I just continue to be in admiration of all of these fantastic science communicators and hope to turn on some light bulbs myself, like Carl Sagan did for me.
And there's so many opportunities for young people to get involved in space exploration. I mean, you have to be an engineer. I'm a geologist. There are biologists involved. There are people who are involved in various studies of the human body. I mean, it's just a whole range of things. Really, no matter what you're interested in, I mean, you can almost find a way to get involved. How did you get interested in planetary protection in the first place?
Yeah, my main interest stemmed, I was actually playing with plasma right? For my PhD work. Actually, before I started my PhD work, as an undergrad, I had co-ops at NASA Langley. And one of my rotations focused on plasma used for levitation and ballistic guidance. And that evolved into, wow, I think plasmas are pretty cool. And so I focused on that for my PhD work. And I was fortunate to be working on a project funded by the NASA Planetary Protection Officer at that time, Cassie Conley. And the goal of that research was to sterilize spacecraft material using the plasma system that I was working on. So that was my natural segue into planetary protection, because the whole goal was to get rid of extremophiles that we may find on surfaces before we launch our spacecraft. So yeah, it's a very unique discipline, but I got a shoe right into the heart of planetary protection.
You have an opportunity in Jezreel crater to look at a place where life may have existed and been deposited in sediment in what's really a harsh environment. Mars never had an ozone layer to protect it the way we have protection here from the ultraviolet radiation. The atmosphere gets bombarded by the solar wind, et cetera. How do you think that life in a place like Jezreel crater might be similar or different from life that we might be familiar with here on the Earth?
Yeah, so the goal of the exploration that we're doing right now is to find signs of ancient microbial life. So as of right now, we don't expect, at least at the depths that we're drilling, to find anything living. But in the past, back when it did have an atmosphere, like you mentioned, not an ozone layer, but it did have some sort of protection, the hope was that something did evolve or stem on the surface of Mars to later be detected by us in the future. But yeah, the goal is to be able to find that sign of life, whether or not it is similar to ours or different. That is one of the exciting things that I want to learn about. Because if we do find, for example, a sign of life, ancient life, then we can ask ourselves, did it evolve independently on the surface of Mars? Did Mars evolve life and seed the Earth? Did the Earth seed life on Mars in the past, and then it went extinct because of the atmosphere thinning and all of that harsh radiation that you mentioned? So there are so many questions. Of course, naturally, as scientists, you get one answer to a question, then you get 50 more questions after that. Right. So yeah, let's see if it's similar to ours or different, or if it even exists at all.
Yeah. And then the last question before we have to wrap up for the night, you think about planetary protection, but is the rover itself going to be protected going forward? We think about these incredibly historic landing sites where these spacecraft, including Apollo landing sites on the moon, and these spacecraft on Mars. I mean, these are incredibly important places for future generations. How are the landing sites themselves and the spacecraft being protected?
That's a fantastic question. Yeah, I don't have insight into whether or not there are going to be landmarks or national monuments set up in the future. I think that's an intriguing thought in the future, especially with the increase in possible commercial applications on Mars. If we have more people, for example, going to the moon, then that's probably a thought that might be accelerated and put into play. With Mars, it's kind of a long debate as far as humans possibly messing it up. I mean, you could send several things to the surface of Mars, and it would be great if it was protected. So yeah, I don't have insight into that, but what a fantastic question, and what a fantastic thought exercise.
Yeah. Well, again, this is the Smithsonian, and that is exactly the kind of thing that we think about in our spare time is how do you protect these incredibly important, right now they're active spacecraft, but in the future there'll be artifacts, and how do you protect those for future generations? So in closing tonight, I want to thank you again very much for this incredibly insightful presentation. It's really been a complex topic, and you've explained it brilliantly. So thank you so much. A big thank you to our sponsors, Aerojet, Rocketdyne, and United Launch Alliance, for making this lecture series possible. Please join us for the next Exploring Space lecture, which will take place on April 27th. The title is Sampling an Asteroid to Understand the Birth of Our Planet, where we will take a look at the asteroid Bennu and what we're learning from sampling that object. Our May and June lectures will focus on Saturn's moon Titan and the planet Venus, which both have very thick atmospheres, but are really very different worlds. In one case, frozen solid, on the other case, the hottest world in the solar system. Please don't forget to sign up for reminders on our website, and please subscribe to our YouTube channel. Thank you again, everyone, for joining us, and good night.